Hi, good day to everyone. My name is Ashreen from Universiti Putra Malaysia. So today I'll be presenting a little bit on my project titled Luminescent Nanoparticles from cocoa pod husks as bioimaging tools. Um, So the luminescent nanoparticles I'm speaking of is carbon dots. So what are carbon dots? They are miniature spherical balls with less than 10 nanometer in size. They are known by many names like carbon nanodots, quantum dots of graphene, and quantum dots of carbon. So carbon dots now are made from various natural precursors. So across many literature, they have used like Manil Kora zapota fruit. In Malaysia, it's known as chiku. We have the guava leaves. They use guava leaves, and even the flower here, Syringa oblata, to make carbon dots. So in Malaysia, one of our major sectors is the agriculture sector. So we would love to use the agro waste to do synthesis of nanomaterials. So the application of carbon dots are various. It goes from antifungi, biosensing, antimicrobial, antiviral, radiotherapy, phototherapy, drug engine delivery, and the one that I want to focus on is bioimaging. So currently they're using gallium, which is expensive, and also there's a radiation dose accumulation. So carbon dots, I would love to focus on this area. So because carbon dots as natural precursors are sustainable and they do not have any harmful effect and they have fluorescent properties. So the carbon dot methods, there are two. There are the top-down methods and the bottom-up approaches. The top-down methods are mostly like from bigger carbon precursors like graphite, and then they break it down to smaller carbon dots. So it is laser ablation, electric discharge, and electrochemical process. So the approach that I'm using is the bottom-up approach, which is we use organic precursors to synthesize carbon nanomaterials. It's very easy. It's a one-step synthesis. The approaches are like in microwave, hydrothermal, and also um, pyrolysis. So the method I'm using is microwave. It is a one-step synthesis. It's sustainable, economical. You can control the reaction parameters easily. So the agro waste that I'm using is known as cocoa. In scientific terms, is Theobroma cocoa, and it's planted in the Americas, in Africa, and in Asia. So annually, it generates a lot of waste. Like from 2018 to 2019, a worldwide waste production from the cocoa industry is about 4.8 million tons. So the main part of the fruit is the cocoa pod husk, which is 75% of the fruit. And Malaysia disposes about like 900,000 kilograms of the CPH annually. So in the cocoa pod husk, actually, there's chemical components like cellulose, hemicellulose, and lignin. And also there's a structure called polyphenols, which is important in the protection against degenerative diseases and also has antimicrobial effects. So the materials and methods, here we have the cocoa pod husk, and then I'll dry it, and then I grind it, and then to fine powder, and then I'll mix it with water, put it into the microwave for a certain amount of time and a certain amount of temperature, and then I will take it out, centrifuge, and then filter, and voila, I'll get my green fluorescent materials, as you can see here under the UV light. So I did characterizations like FTIR, XRD, UVs, and PL. So I went a bit further and tried to do the cytotoxicity test, brine shrimp toxicity assay. So we use the model organism, which is the Artemi nopli, all known by brine shrimp. So I use the 96 well plates. So each well consists of 10 to 15 eggs, and then I'll incubate it for like 24 to 48 hours. So from there, I will um, um, take information such as hatching percentage, lethality percentage, and the LC50. So the first result will be the FTIR, which shows that they have a lot of chemical compounds here, and also have enormous functionalities. So around 3,300 to 3,200, there'll be the OH stretching vibrations, and 1725 and also 1040 here is like a 1725 would refer to the carbon double bond oxygen, and the 1040 would be the carbon single bond oxygen. So looking at the FTR results, they have like presence of a lot of carboxylic groups, so which increases the presence of oxygen on the surface of the carbon dots. So when there's an increase in oxygen groups, which will lead to increase in surface defects. So these surface defects will allow for the trap for excitons to be trapped, giving a rate-shifted emission during the photoluminescence. So, and also the presence of the functional groups actually gives rise to like a very outstanding solubility of the nanoparticles. So in the XRD result, it shows that the at the 2 degree around the 2 theta, like around 20 degrees to 21 degrees, it shows a peak here, meaning that the structure of the carbon dot is amorphous. Okay, so for the UVs, of UVs results, so there are two major peaks at 235 nm, which shows that there is an n-pi electronic transition, and a broad peak around 293 nm, which is a pi-pi electronic transition. So presence of the electronic transition prove that there is an occurrence of functional groups on the surface of the cocopod husk carbon dots. So the UVs is like a blueprint, like it indicates if the method was successful to produce the carbon dots. So in this case, yeah. So also the photoluminescent result shows that it is an excitation dependent PL feature. So something spectacular actually happens when CDs are excited at 380 nm and beyond. Twin peaks actually occur in the range of 380 nm to 500 nm. So, and by these twin peaks, and it's followed by an emission peak at 490 nm when it's excited at 400 nm. So the emission peaks actually begin to decline beyond the excitation of 500 nm. So the wide photoluminescent peaks could be contributed to the effects of quantum confinement or age deformities. So photoluminescent properties actually depend on the different particle size of the carbon dots. So various sizes of the carbon dots will correspond to the excitement of a specific wavelength. So when we look back at the FTIR results, when they have a lot of carboxylic groups, meaning a lot of oxygen groups are present. So when there are a lot of surface defects, and when the surface defect excitons will be trapped, then it is evident and true in the photoluminescent graph that the emission peaks appear right after the 400 nm. Okay, so for the brine stream lethality assay, for the lethality assay at 0.001 milligram per ml, it shows that the brine stream actually survives better than the control solution, which is just the salt solution. It was actually very active and happy, but however, at 1 milligram per ml of the nanoparticles, it had a high percentage of lethality. So over here shows the hatching assay. Actually, as you can see here, at 1 milligram per ml, it shows a very high hatching, but somehow or rather the brine stream acid, the brine stream didn't survive so well. But at 0.01, even though it's a low hatching, the hatching actually increases slowly after 48 hours, and it survives better. So this means that the nanoparticle solution is dose dependent. Maybe the carbon dot solution actually induces the hatching, but the survivability of the brine stream depends on the concentration of the solution. So as you can see here from the picture, at 0.001 milligram per ml, it actually survives very well. It was swimming happily, but somehow or rather at 1 milligram per ml, maybe the dose is too high for it. It does hatch, but it is stagnant in the solution. So it goes into the umbrella stage and then it just dies off. So for the LC50, according to May's toxicity index, if it's less than 1.0 milligram per ml, then it is toxic. So mine is actually 1.32 milligram per ml. So it shows that it is non-toxic in this aspect. So conclusion, so carbon dots made from natural precursors retain their best properties when tested onto different types of cell lines and animal models across many literature. So carbon dots are very suitable to be used in bioimaging, cell tracking, as they do not pose any danger to the surrounding cells of a living organism. And also, okay, so yeah, so future works for my carbon dots that I'm looking forward for is like maybe cancer diagnosis and also cancer therapy. So I would like to acknowledge Kurita Grant and thank Lembaga Cocoa Malaysia, the Malaysian Cocoa Board, for giving me the natural precursors. And also these are my references. Thank you.
Thank you so much, Ashreen. for this thorough work. Very interesting presentation. And of course, there's a lot of interest now in polyphenols, and it's really exciting to know that you can actually generate, using green synthesis, these carbon dots. So I'm sure there will be a lot of application in that regard. It's also interesting how you actually use this brine shrimp for cytotoxicity assays. If I may just start with a few questions. Well, one question initially. Have you tested the cytotoxicity on cell types, for example, that relate to your application? So various cell lines, etc.
For now, no, actually, we are in the process of wanting to do it actually, but due to the pandemic, it's in progress. We're looking forward to do that in the near
Yeah.
future. Yeah.
Okay. Yeah, I think that will be really very exciting.
Yeah.
Um, Also, I mean, I guess polyphenols are known for their antioxidant properties. And I'm wondering whether you tested the effect of your carbon dots on the generation of any Ross moieties at all?
Oh no, also in progress because
Okay,
I know that there's Ross and I'm really excited to actually test this, but in the near future, yeah.
okay, brilliant. Um, so if there are any questions for Ashreen, please pop those into the chat and I'll be able to read them out um, to Ashreen. Any questions? Okay,
Like currently, the one that we're looking forward for is to do for colon cancer actually in the future. Yeah,
yeah.
so we need to do that.
Okay, brilliant, brilliant. Well, um, there is one question. Um, for cancer diagnostics, are you relying on passive accumulation at the tumor site?
Mm, um, yeah, I think so. Yeah.
So I guess the question is, are you planning to do any targeted delivery? Uh,
Yes. Yeah. Yep. I want to do for like brain cancer, maybe for drug delivery for brain cancer. So I was hoping to do that is also in progress.
okay,
Mhm.
okay. That's lovely, Ashreen. Thank you
Uh,
so much.